to be here. Ooh. I'm very glad to be here. And what I'm going to talk about today is in silico directed evolution of proteins in the age of AI. So I'm Stefano Angioletti-Uberti. I'm a professor in computational bio-nanotechnology in the material department at Imperial College London. And yeah, and I will, as I said, like today we're going to talk about basically protein design and what does it mean and what does it mean to use AI for protein design. And I will try to give you an idea of how my group in particular use AI for this kind of thoughts. Like I've just been told that more or less your background is pretty varied, like people from math, physics, computer science. I guess maybe not many people from biotechnology or bioengineering. So I might have to explain some concepts related to proteins. Um, but yeah, we'll take it from there. Okay, so this is the outline of my talk. I think before we start, I will give you a brief introduction about myself and the Soft Nano Lab. So that's at Imperial College. So Soft Nano Lab is basically the name of my group, the group that I lead. Then I will provide a very quick intro to protein design, trying to describe what does it mean to design a protein in the first place. And then I will start to go a little bit more into details, and I will spend some time talking about how do we simulate effectively evolution guided by artificial intelligence to do this protein design. And this is what I call the Bagel approach. The reason why I call it the Bagel approach is simply because Bagel is the software that we developed in my group to do this type of simulations. And then I will present like a couple of case study on selective binder design and enzyme generation. And I'll try to leave some time at the end for Q&A. So if you have any question, you'll have time at the end to ask. At the same time, please, if for any reason there is something unclear during the presentation, stop me and ask question immediately. Just one thing, I can't really see the screen. I can't really see you. I just see my screen. So if you have any question, don't just raise the hand, just like open your mic and say it, okay? Good. So let me tell you a bit my personal story. So there is a before AI and after AI. So myself, I did my PhD in in the Department of Materials, studying computer simulation of solid liquid interfaces. Once I finished my PhD, and this was like a purely computational PhD, so no experiments, but I was developing theoretical models using molecular dynamics and Monte Carlo simulations to study solid liquid interfaces in metallic alloys. After that, I moved to Cambridge. I was in Cambridge for a couple of years in the group of Dan Frankel, where we were studying a peculiar system called DNA-coated colloids. These are, let's say, nanoparticles that are coated with little strands of DNA. And they can be used for different things. At the time, we were not necessarily interested in any specific application, but more into understanding how the system works on a very general level. After Cambridge, I actually moved to Germany. I was there for about 3 years with a Humboldt Fellowship hosted by Joachim Zubiel and Matthias Ballav. And this is where I started to work on nanoreactors and proteins and the interaction between proteins and nanoparticles. So effectively, the first time that I started to do anything related to proteins was about 2012, which it's about 14, 15 years ago at this point. I really want to point out that until this point, all the work that I've been doing was purely computational. So it was computational and a lot of also pen and paper theory. 2015 to 2022, 2015 to 2016, I actually spent 1 year in Beijing, in China. And then I moved back to Imperial when I got a position there. And I started to do computational design, but focused on studying nanoparticles still, but for drug delivery purposes. And basically 2016 is when I moved back to Imperial and I started to build and lead the Soft Nano Lab group. And then 2022 is when I started to introduce AI, or what I would better categorize as deep learning, let's say, models to my computational workflows. And from there's been quite an acceleration. So 2022, that's when I started to use AI. 2023, we spun out a company from my group, Nanograb. This was a company that basically combined the previous work that I did on targeted delivery using nanoparticle with, let's say, a bit of technology that was only made possible by the use of AI. So before that, I would have never been able, with the work that I previously did, to build this technology, but that was the first thing that was enabled by AI in my group. 2024, another spin out, we spun out Aminoanalytica. Aminoanalytica is basically like an AI protein design agent. And we started, 2024 is also when we started the first experimental validations of AI design. 2025 is when we finally came out with this software bagel that I'm going to extensively talk about. And we had additional experimental validation for AI design for cars, which is a system I'm going to briefly talk in a second. So right now, the Soft Nano Lab focuses broadly in everything related to AI for protein design and molecular design. So not really just protein, but in general, molecular design. And there are four main areas that my group focuses on. One is the use of the design of peptides and protein-based binders for targeting. Like I'm going to specify what I mean by binders in a second. The other part is related to enzymes for industrial catalysis and for therapeutic application. Uh, just to be clear, enzymes is just a different word for, or it's not just a different word, but enzymes are proteins, but there are peculiar proteins that can catalyze a chemical reaction, but they are still protein nonetheless. Part of my group also works on cars design. Cars is not like our car, but car is short for chimeric antigen receptors, and these are basically, let's say, these are engineered, effectively they are engineered cells of the immune system, where the engineering part is at this point here, where basically what we design is we design binders that allow the cell to recognize another cell and kill it. So in this case, typically CAR therapy is used for against tumor. And then, like a fourth branch of what we're doing in my group, it's actually working on methods development. So not only on specific application of AI, but also on developing new AI deep learning based methods. I'm talking about PLM. So PLM stands for Protein Language Model, and software for, let's say, AI enhanced design, like effectively like like Bagel here. Okay, so protein design, what does it exactly mean? So to design a protein means to find a sequence of amino acids that, first of all, folds into a specific structure. And then, because it folds in a specific structure, this structure embeds the protein with a certain functionality. Okay. For example, I can have a sequence of amino acid, okay? And keep in mind that a protein in general is just think about a protein as a polymer, so a linear connection, like a string of amino acids. Amino acids are just specific type of molecules. This polymer, unlike, let's say, normal polymers, what it does is that it can fold in a specific structure, okay, like the one drawn here. And then typically this structure can have certain functionality. For example, in this case, what you have in purple here is another target protein. And because of the shape of this green protein, they are complementary to each other, and they can actually bind to each other. So for example, you can use something like this if you want to have a diagnostic test, for example, that needs to detect the purple protein in solution. You can design a binder, okay, and then stick it on top of a lateral flow test. And then when the protein is in solution will stick to the binder, you will have a signal, and that's how you can do, for example, diagnostic detection. Okay, now I'm talking here about functionality. So what does it mean to achieve a target functionality? Typically, there are two classes of functionality when we talk about protein design. The first is what I would generally call binders. Okay, a binder is a protein that can selectively bind a user-specified target epitope. An epitope is just a part of another protein or a part of another molecule. And what do you use them for? Well, you can use them in different ways, but one typical use for binders is to is to work as agonist or antagonist drugs. An agonist drug is just a drug that mimic an existing protein in your body. The existing protein normally activate a function. An agonist protein would be some protein that can activate that function even more than the natural counterpart. At the same time, an antagonist drug would be something that, by binding to the same receptor that your protein does, prevents that protein from binding and it stops activation. So, for example, an antagonist drug would be an antibody that can bind to a protein, to a membrane receptor on a cell, and because the membrane receptor is what used to receive the signal from the outside to activate a cell for certain function, the cell stops working. While you can use protein binders as drugs, you can also use them purely as diagnostic and targeting agents, and you can use them, for example, for CAR-T technology and many other uses. Okay. The other way in which, or better, the other functionality class that people talk about when talking about proteins is enzymes. And as I said, in this case, what we want to do is we want to design a protein. Okay? So we want to design an amino acid sequence that, because of that exact sequence, folds in a specific structure. And this specific structure contains one internal structural motif. Okay? So for example, one pocket that provides the right chemical environment to start to catalyze a reaction. So the example I'm putting here, and we see it later as well, this protein is formulase, okay, here. And then here in this region, what happens is that 3 molecules of formaldehyde would come into this region, a reaction occurs, okay, and you get a product that is different from the original one. In this case, it's just like an isomorphic reaction. Typically, what happens for catalysts just, let's say, enhance the speed at which a reaction occur, but don't really, are not consumed. So it's not like a part of a reaction itself. The catalyst is just, let's say, an enabler for that reaction. Okay, so protein design. There are many different ways in which we can do protein design. And for the sake of simplicity, I try to make them into 2 categories. The first category is generative approaches. Okay? In a generative approach, you can train a deep neural network to find a new optimal sequence from scratch in a single step. So this is the so-called zero-shot prediction. Generative approaches typically are based on, they can be based on various types of deep learning models. So typically we see autoregressive model. For example, ChatGPT is an autoregressive model, but you also have diffusion models. Diffusion models are those, for example, that are used in applications like Sora to make videos or to make pictures from text. Now, in order to train generative model, you typically need quite a large database with specific examples of the quantity that you need to optimize. What do I mean by that? What I mean is that if you want, for example, to design a protein binder, you need to have a large database of protein that binds to a specific target. So if you have that large database, then you would be able to train your generative model. But you need to have a database that contains already, let's say, examples of the quantity that you want to optimize. For this reason, if you wanted to do multi-objective optimization, this is not trivial. Okay? This is because the generative step is typically conditioned to optimize a single quantity. And if you want to optimize multiple quantity at once, you typically focus the generative part only on optimizing one. And then downstream, what you do is you take all the molecules, all the proteins that you generated, and then you filter them based on the other properties that you want to have. The problem with this approach would be that, for example, you might end up generating millions of potential protein candidates, all that have the property that you optimize for, but that lack any of the other property that you want to use as filter. So eventually, and especially if it's a very hard multi-optimization problem where, for example, what the quantity that you're trying to optimize for can actually is, let's say, suboptimal for some of the other property that you want to like to optimize, then I mean, this case where you can't really filter anything because none of the protein would pass the filter, it's relatively common. Now, the advantage of the generative approaches is that at least for protein design, they actually have a very strong experimental validation. So if you want to make proteins that bind to other protein, the success rate measured in terms of how many of the proteins that are generated are really confirmed by experiment, at this point are basically orders of magnitude better than what we were able to achieve before the use of AI at all. Okay, second type of approach is what I would call directed evolution. Okay? In directed evolution, what you try to do is to simulate evolutionary pressure. What does it mean to simulate evolutionary pressure? Well, if you look in nature, you might have a protein. The protein, let's say, has a certain functionality, and because it has a certain functionality, the organism in which the protein exists survive. If now you have a mutation inside that protein that makes the functionality of the protein better, for example, it allows the organism to use more oxygen or produce more energy than before, then that organism will typically be able to reproduce or for a longer time, and which means that basically evolution will actually select organisms that produce that protein, and then mutation by mutation by mutation, the protein at some point will be optimized. This is kind of like how evolution optimized, optimized and keep optimizing everything in the natural world. So directed evolution as a general method is something that it's relatively old. It's a classic method. It long predates AI. It's not something that we were not able to do before AI. We just needed to use a slightly different model. Directed evolution in silico, it belongs to the class of so-called energy-based model. So you have an energy, or you can call it energy, like you can call it a loss, or you can call it a fitness function. They are all synonyms. And this energy guides evolution in multiple steps. How is this evolution guided? In your computer, you basically start with a known protein, or you start with a random amino acid sequence, you make one mutation, and then you accept or reject that mutation based on whether or not your loss function has increased or decreased. And you keep doing it until you reach the desired protein. Obviously, compared to a generative approach where you basically ask AI to generate immediately a protein with the right property, this is a sequential approach, and it can take a long time before you can find any minimum. At the same time, the advantage is that you can, in a directed evolution approach, you can use multiple energy terms. So I can make my energy, my loss function, my fitness function, I can combine different parts. So I can have one part of the energy that, for example, looks for proteins that binds better. One part of the energy that looks for protein that are easier to produce. One part of the energy that accounts for other properties that I want to embed in my final design. And because I can consider them at once, doing multi-objective optimization, it's actually really easy. So this is the big advantage compared, let's say, to a generative approach where this is not so trivial. Okay, so based on this, the choice we took in my group, at least, is that we do protein design using this directed evolution. We believe that we have a higher control on, like, for example, we can easily conserve a specific region of a protein, which might be something important in order not to lose certain functionality. It's an approach that actually, instead of trying to generate protein from like a complete sequence from scratch, you can always start from a solution, a suboptimal but known solution that is found in nature. So it's always easy, let's say, to do optimization. It's trivial and multi-objective, as I was describing before. It's modular, so we can split the energy in different terms. And I think one of the most important things is that the directed evolution approach, at least in the way we implemented it, it's actually time-proof. What I mean by time-proof is that if a new model comes up tomorrow, if a new AI model, for example, that can tell us something about protein comes up tomorrow, I can just do directed evolution using this new model. Whereas in a generative approach, like basically each model substitute the one before. They are not interchangeable and that are not interusable. Okay, so you might wonder, okay, so until now, we discussed about directed evolution and so on, but where is the AI in a directed evolution approach? Well, we typically use AI or deep learning, or what I would call deep learning based algorithm, in two ways for protein design. One is protein folding models. The protein folding model is a deep neural network that takes as input an amino acid sequence and outputs what is the structure of that molecule, of that sequence. The other type of AI-based model that we use are so-called protein language models. So in a PLM, you input the sequence of amino acid corresponding to the protein, and the model outputs a compressed description of all the important information inside the sequence. Now, this is important because with this compressed description, we can build standard machine learning model using as input these quantities. Because it's a compressed description of the protein, we can actually put on top of protein language model very, very simple machine learning model, even linear regression model, and still abstract, very, very important, I mean, very useful, let's say, not just very useful information, but very accurate information from the output of this protein language model. In practice, okay, artificial intelligence in a directed evolution approach is used to evaluate the different energy terms that end up in the model. So, for example, you might have structural terms, okay? So I might need to know the structure of the protein because I want to know which part of two proteins interact with each other. What I can do is I have a target, I have the sequence of the target protein, I have a sequence of the proposed binder, and I can query a model like AlphaFold and ask to AlphaFold, please, can you give me the structure of these two proteins when they are in contact with each other? And you get out this structure, and then based on this structure, you can use other models to evaluate what is the interaction energy between the two proteins. On top of this, these models, and we're going to see an example later, provide quite informative confidence metrics. So AI, typically this AI-based model, they will tell you they not only will give you an answer, but they will also tell you how accurate or how confident they are in the answer that they provided. The other type, as I said, like the other way in which we use AI is by basically we use it to get general sequence property function. What I mean is, what I was trying to describe before is that based on the description provided by a protein language model, we can make very accurate prediction of many quantity of interest directly from the sequence without passing from a structural prediction. So in this case, for example, I can provide the sequence of this protein here, and I can query to AI, and I can ask, okay, is this protein soluble? The protein language model will give me a compressed information inside the sequence. It will pass this compressed information to another machine learning model, and then the model can tell me, okay, I'm 90% confident that this protein will be soluble and 10% that it will not. And I directed evolution. Okay, so this is the way we use AI. So as I said, to evaluate different energy terms. In the future, everything that we do in protein design will actually be direct. I mean, the whole, let's say, optimization pipeline will be directed by AI using agentic AI. In practice, this means that AI will be able to choose which question to ask to aid optimization, which truly means AI will be able to choose the right energy term to optimize a protein so that it has exactly a certain function. It will be able to set up the simulation directly, and it will even be able to send the output directly to robots for downstream experimental testing, get the output of these robots, and use them automatically to iterate on the design. And this is exactly what we are already doing with a company called Amino Analytica here in London, which is working exactly on this type of problems. Okay,
Prof,
now, yes.
on the previous slide, well, not only the previous slide, but I'm querying
Yeah,
more light on the energy terms. Like when you say energy terms in order, it sounds abstract, but somewhere you highlighted on the when the peptides or the binder was binded with the protein sequencing.
yeah.
And I'm wondering, are we looking at the energy terms in terms of how the binder is relating with the protein, or we're looking at energy terms when it comes to model optimizations in
If
that
I,
sense?
yeah, so I would say, let me finish this part of the presentation. Then I will give you some example and we can go back to this question later.
All right.
Yeah, because I
Oh,
think the example will be, with some example, it will be much easier to understand some of the concepts that I just talked about.
sure,
Yeah,
sure.
and if not, let me know and when we can discuss it again.
All right.
Okay, so let me go a little bit more into the details of how we do directed evolution using our code Bagel. And by the way, I'm putting here just um, as a reminder, let's say, so the code that we developed, it's fully open source. It's free for use for everybody, like doesn't matter, academics, commercial. So if you want to have a look at the code, you can just go on GitHub on our page. It's public. You can download the code. And if you find it useful and you want to contribute to it there is a series of, let's say, pull requests and issues on our GitHub page. So if you want to help us, you're more than welcome to do it. Okay, now we talked about directed evolution. So how does it exactly work? Okay. So directed evolution, you can look at it from a physics perspective. Okay, you can look at it as sampling of an energy landscape. What does it mean? It means that by deciding the energy terms that end up in our optimization problem, and because the energy terms depend on the sequence, okay, we will have an energy landscape, meaning that we will have an energy that where each point in this, let's say in this two, this is like a two-dimensional representation, but each point in this two-dimensional representation actually correspond to a different sequence of amino acids. So it would correspond to a different protein. It will also have a different energy. So what we do, okay, is we start from a point that can correspond, as I said, either to an initial sequence that it's somehow from a protein that we know it's close to the solution we want, or you can start from scratch, that doesn't matter. But what we do is effectively we do a random walk or a semi-random walk directed by the energy. Okay? So you can start from this point here. So this point here corresponds to this sequence here. And then you can start to do mutations and you go from one sequence to another to another. And effectively at each time, you use the energy to evaluate whether or not your energy is going up or down. If the energy goes up, you accept it or reject it with an exponential function. If the energy goes down, you always accept it. And then you keep running this algorithm until you find effectively the minimum of your energy function. Now, this approach, in a way, combines a bit of the best of AI with a classic approach. Why? Because we use AI to evaluate the different energy terms. Okay, but the sampling approach, the optimization approach itself, it's just standard Monte Carlo optimization. So we build on a solid mathematical background. So standard Monte Carlo has been known for Markov chain Monte Carlo as a mathematical technique has been known for more than 100 years. Computationally, we've been using Monte Carlo, Markov chain Monte Carlo since the 1930s, 1940s. So it's 80 plus year of accumulated knowledge. And there is a lot of known solution and heuristics to problems and recipes that we can use in order to speed up, let's say, the directed evolution process. And it's conceptually very easy to understand and extend. So every physicist that have done a bit of statistical mechanics would know would have a good understanding of how exactly this works. Now, how do you set up a protein design problem? Okay, so the first ingredient, okay, you need to decide, I mean, the basic ingredient is a chain. A chain is just a sequence of amino acid. And in this sequence of amino acid, you might decide that the whole sequence can be varied, or if you want, you can decide that part of the sequence should be completely conserved and immutable. And the software will take it into account during the mutation. It will never mutate any of the amino acid corresponding to the mutable part. Now, on top of a sequence, you will need to decide what are the different energy terms. There are energy terms that depend on the overall system. We can call them zero body energy term. There are energy terms that depend on single amino acids. And then there are energy terms that depend on pair of amino acids. So we can call them like two body or one body term. Effectively, energy can be associated to any part of the amino acid chain that you have in the system, or they can also be associated to multiple chains. Let me give you an example. For example, if I have to design a protein binder, what I can do is I have my sequence chain A, and I can say one energy term is the energy of interaction between the amino acid A1 to A4, which correspond, let's say in this case, would correspond to this region, with the interaction with what? With the interaction only of the amino acid from B4 to B8 that would correspond to this region. At this point, if I do something like this and I let my Monte Carlo algorithm go on, it will change the sequence of chain A, assuming chain B is your target, so we don't vary it, but it will change the sequence of AA, the sequence of chain A. And at each point, it will evaluate the energy of interaction between all the part here, between A1 and 2A4, and all the part here before 2B8, and it will try to increase the strength of this interaction. Or, in other words, it will try to decrease the binding energy of the interaction between these two parts. And eventually, you would get what we call the bind. Now, an important thing is to take into account that for an optimization problem, you don't necessarily, I mean, sometimes you have a single chain, sometimes you can have multiple chains. For example, in this problem here, you have two chains. The combination of multiple chain, each of which with the energy term associated to them, it's what we call in our software, we call the state. So the state is a collection of interacting chain and their associated energy. And finally, we can have multiple states. And I'll give you an example later of a typical multi-state optimization problem. So the total energy is the sum of all the energy of the single states that you have in your system. With this, we can specify basically almost any optimization problem that we have in protein design and protein engineering. Now, once you have, let's say, your system has been specified, then you need to specify which oracles you use in order to, let's say, evaluate the energy terms. What I call here oracles are just mathematical models that connect the sequence of the chain to the final energy term that we want to calculate. For example, one energy term can be associated to the structure of the protein. So what we can do, one oracle would be an algorithm like AlphaFold that, given the sequence, gives you the structure, and then we are able to evaluate that energy term. In general, keep in mind that energies are not necessarily what a physicist would call an energy. You can think about energies here as truly any quantity that you want to minimize in the optimal design. You have a system, you have specified which oracles, which AI model are necessary in order to calculate the energy of the system. You also have to specify what a mutation protocol. I was saying before that a mutation protocol could be something whereby you take one amino acid at a time and you just mutate that amino acid and you evaluate the mutation. But in principle, you can think about different mutation protocols. For example, you can have canonical accommodation protocol, you only substitute one amino acid with another amino acid, but you can also have a grand canonical protocol where actually you also allow to insert or delete amino acids, or you can have many other type of protocols where instead of inserting or deleting or changing an amino acid, you can combine like blocks of different amino acid chain. You can implement mutation protocol as you want. It doesn't really matter. Each of those can be used interchangeably inside, let's say, our algorithms. Once you specify all this, the software takes care of the optimization for you. So it's like effectively specifying an optimization problem just means specifying some initial chain, specifying the energy terms and the states, and that's it. Once you do that, then the software will do the optimization, and at the end of the optimization, you will have, let's say, your design protein, and that's it. This is just a summary of how the optimization effectively works. So we start with some chains, some initial sequence, and compute the system energy. You mutate the chain, you fold the chain or embed with oracles across states. So basically, you compute the system energy, and then you accept or reject based on a Monte Carlo acceptance criteria. And that's it. It's nothing more than this. So this part is extremely simple. And then you keep iterating between point 4 and point 2 until, let's say, you reach convergence. Convergence just means that at some point you will see that the energy of the system will stabilize, will more or less plateau, and that's it. At that point, you stop. Okay, so let me talk a bit about applications that we solved using exactly this software. The first application is the design of selective binders. So the problem statement is the following. So currently, most type of, let's say, generative AI for binders, they truly only optimize for binding strength, but not selectivity. What I mean is that typically people optimize to find a protein sequence that binds as strong as possible to a target, but does not care necessarily about whether or not the same design protein will also bind to other proteins. And this is fine for certain application, but it's but it's a big problem for other, especially for therapeutics. For example, if you design a protein to bind to a cellular receptor, but then this protein, instead of binding only to its target, it binds to every single cellular receptor, you might actually be blocking important vital function for the cell and eventually kill the cell. And you really don't want that. So what you need to have in this case would be to have a binder that binds the right target and doesn't bind to any other protein. If you think about it, this is exactly the perfect example of a multi-state optimization problem. And it's multi-state for the following reason. So I'll give you the example that we treated in this paper is we have two protein, EGR1 and ZNF, and we want to have a binder. So here, this sequence in blue, we want to have a binder ideally that binds to EGR1, but does not bind at all to ZNF because it would be toxic. Okay, so how do we do this? So you prepare a system with two states. In state one, you just put the two chain, the chain of the binder, B, and the chain that correspond to the target. In state two, you put the chain of the binder and you put a series of chains, NT. So NT now is not a single chain. It can be 1, 2, 3, 4, 5 chains corresponding to other proteins that you do not want to bind. Okay? Now, the energy for state one is the binding strength between your binder chain and your target. The energy for state two, we take minus the binding strength between A and B. So if we do that, and we combine the total energy of the system as the sum of the energy over a different state, what we end up with, if we minimize the energy, is exactly finding a binder, so finding a chain B that will maximize the strength of binding between B and T, and minimize the strength of binding between, sorry, there is a mistake here, and minimize the strength of binding between B and any NT, so non-target sequence. Maybe related to the question or that I think you were asking before. So what is the energy of binding in this case? Well, there are potentially different ways to evaluate the energy of binding between two sequences in this case. What we use for optimization is we don't really calculate the physical energy of binding between the two proteins, but we use the so-called predicted align error. So this is a measure, this is actually a measure of the confidence of the model, okay? And it tells you how strongly correlated are two pair of residues. Now, because two residues that are strongly binding, they are strongly correlated in their movements and in their position, then effectively the predicted aligned error is a measure of the binding strength. It's not exactly the binding strength, it's a proxy for the binding strength, but we can use it as if it was a measure for the binding strength. And what I'm showing you here is exactly what happens during your Monte Carlo optimization algorithm. So the interface predicted aligned error between your target protein and EGR1 keeps decreasing as much as possible, whereas the energy for between your binder protein and ZNF remains very high. High energy is associated to very weak binding, and the low energy is associated to very strong binding, which is exactly what we wanted. And here you can basically see, let's say, the so-called PAE matrix that compares that basically gauges the interaction between the residues of your binder, which are the residues between 0 and about 20, and all the other residues, which are the residues corresponding to your target protein. And as you can see, there is a very small predicted align error between the target, the binder, and the target protein, EGR1, and there is a very high predicted align error, so a very low energy of interaction between the residues in your target and the residues that correspond to the non-target protein, which is exactly what we wanted to achieve. Okay, now another application where it's still about binders, we use similar techniques to design binders for so-called intrinsically disordered epitopes. What is an intrinsically disordered epitope? So proteins typically fall in a rigid structure, but there are certain proteins, there are certain parts of proteins that are not rigid, and they are pretty much disordered. They are very difficult to target. So what does it mean they are very difficult to target? It means that it's difficult to find a binder that would bind to an unstructured region. However, in terms of, let's say, for pharmaceutical application, it would be extremely useful to have binders for unstructured regions. In part because they're about 30% of the human proteome, so of 30% of all the all the proteins in humans consist of intrinsically disordered regions. So if we cannot, basically it means that if you cannot make binders for this region, there are 30% of potential target that can be used for pharmacology are not usable. And this is the problem that we're trying to solve. So how do we solve this problem? Well, background idea is the following. So when we ask a model like AlphaFold, okay, to fold a protein given the sequence, as I was telling before, the model not only gives you a certain structure, but it will also tell you how confident the model is in the structure that it provides. In this case, for example, you can see that for this protein CMYC, which is a transcription factor, there are some regions that are described with very high accuracy, so very high PLDDT, and then there are certain other regions where the PLDDT is very small. Now, one thing that has been noticed in the literature is that when the model has a low confidence, it doesn't necessarily mean that the model is giving you a wrong answer. Often it means that region is an intrinsically disordered region. An intrinsically disordered region is very dynamic. It keeps moving, it keeps changing. So the model cannot tell you cannot give you any structure for the simple reason that that region itself is unstructured. In other words, what I'm trying to say is that we can use the PLDDT, which is a confidence matrix of our AI algorithm, to measure whether or not a region of a protein is a disorder region or it's not a disorder region. Now, the idea then is the following. So what if some proteins are known to co-fold together? Co-folding means that the protein taken singularly, they are unstructured, but when they are close to each other, they can start to form an ordered structure. And this is something that is, this is something that has been observed in natural proteins. We're not inventing anything. But we are going to combine the PLDDT with this idea. And how do we do it? Well, basically, we can use the PLDDT, or better in this case, minus the value of the PLDDT as an energy term. So if you think about it, if you have a disorder region, the disorder region will have a very low PLDDT, and the energy is relatively high. However, if we can find a a binder sequence that went together with the sequence that we want to target, will actually increase the PLDDT. So it means that the binder will co-fold with the region that is initially disordered. Then we can basically use the PLDDT value in order to find actually sequences that will co-fold together, because maximizing the PLDDT will naturally, the PLDDT purely of the initially disordered region means that we can find a binder that will start to structure the region more and more, and eventually generate a binder. And this is exactly what we have done. We take the PLDDT, we use that PLDDT as the driving force for our algorithm. We keep on changing the sequence with directed evolution, and eventually we come up with sequences that indeed structure the initial disorder region, and these are our binder. After that, what we did is we did an in silico validation. What does it mean? Is that what it means is that we use molecular simulation techniques. So this is not AI anymore. These are molecular simulation techniques like molecular dynamics, free energy calculation. And we calculated the physical free energy of binding between the binders that we designed and the target protein. And we calculate these energies, and we get energies of a few tens of kBt, which in binding, which basically prove that our binders that have been designed purely to increase the PLDDT really do bind to the target protein. Okay, final. Do I have time? Maybe 5 minutes, and I'll give you the final application. So I told you, like, there are typically two categories of functionality that we want to improve in protein design. One is binders, and we'll just be talking about it. The other is enzymes. So how can we use it for enzyme? I'll give you a specific case. So in nature, many enzymes are not made by a single chain, but they are made by multiple protein chains that come together. And that's perfectly fine in nature. The problem is that if you want to use this enzyme for industrial use, the enzyme to be functional need the chain to self-assemble into a specific in a specific structure. And sometimes it's difficult for the enzyme to self-assemble in a specific structure if it's not exactly in the condition that it's found in nature. So if you want to use it for industrial catalysis, where you might want to do your reaction at higher temperature compared to what the enzyme would do, let's say, in a natural environment, that's not going to work. So what we are trying to do here is we're trying to use a directed evolution approach to find, let's say, a fused monomerized version and miniaturized version of an enzyme. Okay. So let me show you how we do it, and for a specific example. So this is formulase. Formulase is an enzyme that converts aldehyde in a C3 compound. C3 compound is just a compound that it's an alkylic chain of 3 atoms that contains at least 3 atoms of carbon. Now, in formulase, okay, the catalysis, so this reaction occur exactly at the interface between different chains. So in formulase is a tetrameric, so you have 4 different chains that come together and form this beautiful structure. The catalysis occur exactly in this region described by these rectangles here. So how do we do it? Well, we do the following. We take this region, we take the interface region, and we extract it. So this will be extracting it just means literally that we take the two different chains and we just take the amino acid correspond to the region at the interface. We stitch these two chains that are physically separate, we stitch them together with a so-called linker. A linker is just another piece of another sequence of amino acids. And then the fact is, if you take this, let's say this initial chain, it will not, I mean, if you take this monomerized version initial chain, it will not stay like this. Like you took a piece of a protein, okay, and you're trying to, I mean, you took a piece of a protein, you extracted it from the real protein, it will not fall back exactly to the original, to the original version, because you removed a lot of the other parts of the sequence that are necessary in order to template the structure. However, we can take exactly this region here and basically do a directed evolution approach whereby we mutate the sequence, and each time that we make a mutation, we refold the sequence and we ask how close is the structure of the sequence to the original one that you find in the tetrameric enzyme. And again, we can use basically how far the structure is from the tetrameric enzyme is exactly what we can be measured by the RMSD, the root mean square deviation from the original structure. And we use that as an energy term. So the lower the energy, the closer the sequence will fall to the original structure. And we keep on doing it. And again, we can optimize effectively a monomeric multivariant, a monomeric variant. These are some of the results that we had. Like once we have a monomeric variant, we can actually, let's say, use different type of AI models. So to ask the same question, all the different type of AI model that we use provide us with exactly the same answer, which give, or at least very similar answer, which makes us confident that indeed the answer that we get is correct and not purely dependent on the type of model that we're using. But again, more than that, we can do an in silico validation where, again, we use molecular dynamic simulation. So we take our design sequence, we put it in a molecular model, we do molecular dynamics, and we look at whether or not the sequence that we design will be stable or if it will start to unfold. And if you look at the molecular dynamics results, there are some variants that are actually unstable. For example, the blue and the yellow variant clearly are starting to drift. But then there is a red variant, which has relatively high fluctuation, but remains overall around 0 in terms of the distance between the two parts, which is exactly what you would have in the original enzyme. The original enzyme, so the original multimeric enzyme is the black curve. And as you can see, which basically is stable, and the red curve is stable as well. Here, what you're observing is simply the fact that the original enzyme not only is stable, but it's also the fluctuations are relatively small. In the case of the red enzyme, there is slightly higher fluctuation, but they still remain contained around 2 angstrom, which is, let's say, a decent result. Next step will actually be experimental validation of all of these results. Okay, future direction and how to contribute. So as I said at the beginning, Bagel is an open source package. It's free for academic and commercial users. Please go on the website, have a look at the code. If you want to contribute with your coding skill, you're more than welcome. There is a series of GitHub issues that you can contribute to. Basically, we'd like to implement multiple folding oracles, different type of minimizers, different type of mutation protocol. There is a lot of work to do. And Obviously, if you're interested in experimental validation of some of our design, we are more than happy to talk about it. Finally, let me thank all the members of the software lab, present and past. I'm gonna put picture only of people who are currently in the group because otherwise it would be too many. These are the people who did most of the work that I presented. So I really want to thank them. And I want to end up by thanking also ARIA and Code AI and the ERC, European Research Council, for funding that allowed me to do this research. And I'll take questions now. I'm finished.
Thank you, Prof. There is a question. I want to read it for you.
Yeah.
Ben is asking, have these approaches been applied to neurodegenerative diseases to restore or replace lost or dysfunctional proteins? If so, what level of clinical or preclinical success has been observed, particularly given the complexity of neurosystems? Additionally, do you
Mhm.
see potential for extending this strategy toward organ development for transplantation or even modifying disease progression in chronic conditions such as type 2 diabetes?
Okay. So let me the answer to this question is, let's say, is quite complex, because what we're doing using this approach, it's purely designing proteins. Okay. So if you want to use this approach, for example, to look into neurodegenerative diseases, which is something that people did, it means that first of all, you need to know somehow something about the mechanism of these diseases. And then if something in the mechanism of these diseases contains a protein, or a protein plays a role in some of these diseases, then you can think about using such an approach that I just described to try and find a cure. I'll give you a specific example. So if you look at, for example, Alzheimer starts by, or at least this is what most clinicians still believe, Alzheimer starts by the formation of aggregates of proteins. Typically, there is a protein called tau that, if you look at this encephalogram of a person that has Alzheimer, you see like giant clumps of this protein. So there is a lot of research trying to understand whether or not we can prevent the formation of these clumps. One idea that people floated is exactly to have, let's say, an antagonist for tau. What would mean? For tau to assemble, you need many proteins to bind together, right? So what people are trying to do, for example, is to find a protein that binds to the interface that tau normally would use to bind to other tau protein, and saturate this interface. By saturating this interface, then tau would not be able to attach to another tau protein, and you cannot form aggregates. So if you do something like, so if you rephrase the problem in this way, which which means I have a disease. I know that the disease depends on this protein. I will try to modify the interaction that of this protein with another protein that causes this disease by inventing an antagonist protein. In this case, yes, you can use this type of let's say you can use this type of techniques that I just described to do exactly that. But you need to know already that, you need to know something about the mechanism, and you need to know which protein are, let's say, part of this mechanism, because if you know which proteins are part of this mechanism, you can try to control, let's say, the interaction of these proteins by inserting some protein that disrupt the interaction. And this disruptor is something that you can engineer using directed evolution or using, in general, protein design.
Okay.
Having said that, I don't think, all of this, you have to consider, all of this is quite new. We're talking literally about maybe 4 or 5 years, not more. Actually, probably even less. So in terms of, there is not yet any clinical validation for a concept like that. You have papers that have shown, let's say, that you can in vitro, you can control cellular interaction. There are papers that have shown that you can design protein binders that can then be put in vivo in mice, and they can help restore some certain functionality in disease model? Yes. There is no clinical validation yet on humans for any of this, but it's simply because the field is still in its infancy.
Okay. Prof, thank you. I think my question is about the algorithm for Bagel itself.
Yeah,
And you highlighted that it is literally sampling an energy landscape.
landscape. Yeah,
And the key idea is you're trying to find the low energy where it is sort of like pivoting towards.
yeah.
And my question is, is that the same pattern that is expected even for these intrinsically disordered um, peptides or proteins that we have discussed? The algorithm is still going to behave the same way.
Yes. So let me, I think I went over that example a bit quickly, and maybe, I'm not sure if I was able to explain it accurately enough. But basically, the important thing is the following, is that every single protein design problem, every single optimization problem truly depends, the solution only depends on finding the right energy terms to describe this problem. If you find the right energy terms, then like optimizing it becomes easy. Now, for the case of the intrinsically disordered protein, effectively what we call the energy is just a function that tells us how disordered is a certain region of the protein. So there is a quantity which I said here, it's the so-called PLDDT. The PLDDT is a measure of how disordered a region is. So if the PLDDT is high, the region is not disordered. If the PLDDT is low, the region is disordered. So if we use minus the PLDT value as an energy, and we try to minimize the energy. So minimizing the energy will mean maximizing the PLDDT. When is the PLDDT maximum? The PLDDT will be maximum when the protein that we want to target with its disorder region, together with the binder that we are designing, will start to form, we will co-fold together, okay? Because by co-folding, the PLDT goes up. The PLDT goes up, the energy goes down. And that means you will have found your solution. Keep in mind, the PLDT is literally just a function, okay? It's the predicted local difference test. It's just that you can think about it purely a function that every time that you use an algorithm like AlphaFold to fold the protein, AlphaFold will give you the structure of the protein, and then it will give you a value for each amino acid of the protein that tell you how likely is that amino acid structure to be exactly the one that I gave you. And this is what the PLDT is. It's purely a confidence metric that comes from the algorithm itself. It's not a real energy, but because it's correlated to the energy, it doesn't matter. If you increase one, you will also, in this case, decrease the other, and that's it. And you can use that. And you can do sampling. You can do sampling, or you can do Monte Carlo optimization, and if you find the minimum energy, you will find the maximum PLDDT, and the maximum PLDDT will mean that you will have found a binder in this case.
Wow, that was quite deep. I think I had missed that sort of insight. And thank you very much, Prof. I want to thank you.
You're welcome.
People who have been involved. Thank you very much once again for saving up your time and really talk to our group.
You're welcome.
This has been quite profound. So thank you very much, Prof.
Good.
And thank you very much to everyone. Yeah, and we meet in our subsequent calls to the members. And a good
Good.
Is it a good evening in UK?
Yeah, thank you very much. Thank you very much, guys, and good luck with this initiative. It's a beautiful initiative.
Thank you very
I really
much.
hope you can learn a lot. Okay, bye,
Okay.
everybody.
Bye bye, sir.
Bye.